I'm Madison. And I'm Mollie Hagen.
This diagram is our muscle contraction diagram, and this box represents a muscle cell, and in this section right here we have the sarcomere.
Okay, so in the sarcomere we have the myosin thick filaments, the actin thin filaments. We have an H zone along the myosin thick filament, and about halfway through the actin thin filaments we have a Z line. So muscle contraction begins.
It begins at the motor neuron, and in the motor neuron's axon terminal there are neurotransmitters, and the neurotransmitters transmit an action potential along the sarcolemma membrane, and it goes down the T-tubule. We have the green represents our T-tubule, and it sends a signal to the sarcoplasmic reticulum. In the sarcoplasmic reticulum, calcium is signaled to be released, and calcium releases from high to low, which is passive transport. And so what the calcium does is it binds to the troponin. These beads right here represent the troponin, and the calcium binds. And when the calcium binds to the troponin, it changes the shape of the tropomyosin. And we have our strings, which represent the tropomyosin.
The tropomyosin will change shapes to reveal the actin binding spots, which is represented by the black dots. The myosin head will bind with ATP and hydrolyze it into ADP in a phosphate group. This changes the myosin head into a high-energy configuration shape. The high-energy configuration will allow the myosin head to bind to the actin binding spot, and this binding is called a cross bridge. When they are in the cross bridge, they will then relax into a low-energy configuration state, and the myosin head will pull the actin filaments and will push them together. These actin filaments will come closer together, but they will not overlap. This stimulates the muscle contracting. The actin filaments move closer together and the H zone disappears. In order for the cross bridge to be broken, more ATP is required to come and break the binding spots, and the muscle will then relax. However, in rigor mortis, since the person is dead, there is no longer a way to regulate the calcium ions, and they will all be released and will bind to the troponin, as shown on the diagram. This will cause the muscle to contract. However, the ATP will be used up while binding the myosin to the actin, and there will be no more ATP left, so the body cannot relax and it will be stiff. This will last for about 2 days until bacteria can come along and break down the binds between the myosin and the actin. Thanks for watching our muscle contraction video.